Hello all, welcome to Polyhipster. In this video, Approach to Endocrinology, we will be discussing how to interpret a case of endocrinological disorder and how do we proceed step by step into identifying, diagnosing, and treating it. So let's begin. Starting with a stepwise approach towards an endocrinological disorder. We first define a syndrome by taking detailed history of the presenting complaints followed by proper observation of the signs and symptoms in the patient. Next, we match it to the gland malfunction. We measure the suspecting gland's output and then we consider various diurnal variations and other modifying factors associated with the fifth step is to define the clinical syndromes, whether it is hypo, hyper, or a U condition. U is where the hormone levels are normal. So, if it is a hypo condition, we test it by stimulating the gland. If the gland is not functioning, then we get a blunted response. An example is ACTH stimulation test. Next, if we are suspecting a hyper condition, we'll do a suppression test which will fail in hormone secreting tumors. An example is dexamethasone suppression test, which we usually do in case of Cushing syndrome. Next step is imaging. Next, we aim to halt the disease progression and finally the treatment for the diagnosed condition. What are the various pathological mechanisms of endocrine diseases? They can be categorized into three types. First, where there is excessive secretion or activity of hormone. Next is where there is a deficiency of hormone secretion or deficient hormone activity. And last is when there is a resistance at the hormonal receptor complex. We see excessive hormone secretion or activity mainly in case of neoplastic conditions, autoimmune, iatrogenic, infectious, inflammatory conditions, or in case of activating receptor mutations. A deficient hormone function is noticed in case of autoimmune, iatrogenic, infectious, and inflammatory condition whenever there is hormone mutation or any secondary enzyme defects, developmental defects, nutritional or vitamin deficiencies, hemorrhage, and infarction. Whereas resistance is seen mainly in case of receptor mutation, whether it is membrane or nuclear receptor, or if there is any mutation in the signaling pathway or any post receptor changes. How do we measure the hormones and its related analytes? So, in our day to day laboratory, we use four major techniques. First, being the bioassay, then receptor assay, immunoassay, and instrumental techniques. Let's begin with bioassay technique where physiological responses specific for hormone is measured by two methods, either in vivo or in vitro method. In vivo method, the test material from the patient, mainly blood or urine, is injected into a prepared animal and target gland response is measured. Whereas in vitro method, we we use culture medium to develop tissues or cell or membrane, and in those cultured cells or tissue we measure appropriate hormone response. Bioassay methods are imprecise and are rarely necessary in clinical medicine. So let's continue with receptor based assays. Here, either the assay is done in vitro with interaction of hormones with its biological receptors, or it will measure response such as production of cyclic AMP when a test sample is added to a preparation that includes receptors and necessary cofactors. The disadvantage with in vitro method is that unlabeled hormones will displace the labeled one. Advantage of this method is that it is more sensitive than bioassay and it will reflect the biological function of a hormone, which makes it an advantage over immunoassay. And also, it has a disadvantage where it is not as sensitive as immunoassay and complexity is more. Next are immunoassay techniques, which are most widely used in our day-to-day laboratories. It is also the method of choice for peptides and proteins. Here, labeled antibodies or immunometric assays are used with non-isotropic labels. They use saturating concentration of two or more antibodies, which are often monoclonal, that are prepared against different epitopes of protein molecules. If we take example of two antibodies, the first one will be attached to the solid phase separation system and it will extract hormone from the serum specimen. Whereas the second antibody, also known as detection antibody, will be linked to a signal molecule or label, and both these antibodies will bind to the hormone in a sandwich pattern. Finally, the instrumental techniques, mainly mass spectrometers coupled with gas and liquid chromatograph, the matrix assisted laser desorption or ionization, which is known as SMARLD, and electron spray ionization techniques. These are the most advanced techniques. Here, they mainly do sequencing of peptides, mass determination of picomole quantity of analytes. Advantage of this method is increased sensitivity, high accuracy, speed, and it also allows simultaneous determination of multiple hormones related to the clinical conditions. One should be aware of various specimen requirements and conditions that should be met before collecting the sample for hormone measurements. These include the factors affecting different hormone measurements. For example, insulin is affected by intake of nutrients. Cortisol levels are affected by the time of day. Parathyroid hormone will be affected by the serum calcium level. Therefore, one should be aware of these things before collecting the sample. Next is to know whether the particular condition requires a stimulation or a suppression test and what type of sample is required to measure a certain hormone. Mostly, samples collected include plasma or serum, 24-hour urine, or saliva. That's all for now. See you soon with more videos on endocrinology and other related topics.